Good morning, muffins. Yep, you heard that right. This nerd here created a new enzyme. So, in the winter semester, I audited this course, Enzyme Design in VR. We study foundational knowledge about structures and basic mechanism of enzymes. And we got to play with VR. Pretty fun, not gonna lie. Students in this course will have to do a capstone project, which is a presentation about an industrially useful enzyme. Now, I am definitely the odd one here, auditing as a master's student in computer science in a room full of biochemistry undergrad students. But since I'm not getting a grade, I get to go wild with my imagination. In fact, the professor encouraged me to try wacky ideas, as he liked to have me inspire the student towards computational chemistry. So my group chose alkane monooxygenase, or alkb for short, a class of enzymes seen in some bacteria that bond oxygen atoms to alkanes. Well, my teammate each need to cite a paper and present it to the class. Meanwhile, I get ready to cook and hopefully not obliterate the kitchen. Lab. Well, what can I say? Unlike others here, I just code. The worst I can do is nuke the boot sector. Wait, fuck, I nuked the Windows partition instead. Alright, so I found this paper about how alkb from this bacteria can remove the fluorine atom from one fluorooctane. And of course, I assume everyone here knows about the environmental impact of fluorine compounds and how difficult they are to break down biologically. So this is kind of huge. In the conclusion, they said, it has shown itself amenable to directed evolution and can serve as a model for bio-inspired catalyst design. And the stars seem to have aligned as this bacteria's version of the enzyme has a sequence and structure already published online. Alright, I'm on board. To optimize an enzyme, we'll evaluate how good it is by docking in Gnina, where we take the protein and a target molecule and simulate them interacting to estimate how good they will bind to each other in real life. And if you are in the field of computational biochemistry, you might have noticed the name. Yes, Gnina is a fork of Smina, which itself is a fork of the famous autodoc Vina. Note that Gnina is originally made for receptor-ligand interaction, where the protein just binds to a molecule for it to trigger a signal. We don't really have better open source option though, so we have to make do. Now, Gnina output looks like this. It goes through different poses and gives you a list of pose scores and affinity. The pose score describes how good the pose is from 0 to 1, and affinity is an estimation of the binding affinity. Ideally, both should be increased at the same time. But I looked around and literature suggested that I use CNNVS, which is a product of the above two metrics, which will be large when at least one is larger and the other is not neglected. And in the spirit of trying new things, I will call my system with one fluorodecane. Since I'm quite new and completely lost, I hit up my student premium subscription of Gemini to suggest what to do. And okay, context here. No, this is not sponsored. In fact, most of the brain cell casualties in the next 5 minutes are brought to you by Gemini. Initially, I thought of making a directed evolution pipeline where, starting with the initial amino acid sequence, we dock the enzyme, calculate the interaction between the atoms, feed this data to an LLM, and ask it to suggest a mutation. Then we can fold it again using AlphaFold 2 on Colab and repeat the process. Now, this sounds cool and cutting edge, right? Where we combine two of the most famous AI models we have to build a pipeline that designs new enzymes. Wow, I'm definitely getting a research award for this. No. It soon turns out that this is just a waste of computational power, as neither models are actually needed. First of all, AlphaFold 2 on Colab takes 15 minutes to fold my enzyme, and if I want to examine 30 changes, it's going to take me a whole day. Meanwhile, computing credits are not cheap, and if I want to do it myself, my RTX 3060 just cannot handle it. Well, I mean, technically, this is still better than having the trees and water wasted on generating webcomic slop. No, you will never be your Taoist immortal Grand Knight Elite Hacker self-insert slop. The only technique you mastered was the technique of falling for Bitcoin scams. So I get back into some back and forth with Gemini. We finally settled on this Python package that can simulate a molecule stabilizing to its lowest energy state. Turns out this is perfect for approximating the conformation for small mutations on a protein with experimentally soft structure. Well, perhaps even better than AlphaFolding it from scratch, as we keep and utilize all the information regarding the 400 other amino acids. Well, why didn't you just tell me there's a quicker way instead of glazing me now? My brother in science, if I needed a clanker that said, "Yes, daddy," to everything I ask of it, I could just go to one of those stores in downtown Toronto. Okay, second issue with the cool kids AIs. Now I got the mutation and refold working. I'm gonna steal my parents' credit card and register for some Gemini API keys. I coded a loop to feed it the interaction data and let it run. And all it does is change a bunch of residues into alanine and further turn them into glycine. And then it crashed on me with API server busy errors. Oh hell no, who the frick lobotomized my Gemini API? Alright, back to asking my unpaid RA aka Gemini chat. Turns out that if you look at the set of all papers related to the topic of artificial mutations, a huge chunk of them are just experimentally changing residues to alanine to examine its impact on the overall protein. Alanine is an amino acid that can be seen as a blank or filler residue, since it does not have any special side chain. This makes it perfect as a substitution to examine how important an amino acid residue is in the function of a protein. Oh, and by the way, alanine is different from glycine, which has no side chain at all, making it way too flexible and will cause an alpha helix to collapse. Yes, this is also what I learned in the course. Since the memory space of LLMs absorbed a huge number of these papers, they will of course be biased to suggest alanine. This is like how if your classification dataset is 98% healthy and 2% cancer, then a model answering healthy to everything would technically be 98% correct. Now with all of that, I end up just designing my own simulation loop myself. Starting with a raw file download from PDB, remove the extra leftover stuff, and relax from the crystallized state proteins are usually in during XMR scan to a natural state using OpenMM. At the beginning of each iteration, we have a reference enzyme. We run Gnina doc and get the output with the best CNNVS. Then we inspect the output from Gnina and select the top N residues closest to the molecule. For each of them, we make a copy of the enzyme where we turn that residue into glycine without refolding the enzyme. Then we dock it in Gnina again. This essentially let us assess the impact of the residue in the binding by comparing the enzyme with the residue versus when it's Thanos-snapped away. Then out of these candidates, we shrink down to top M residues sorted by highest impact. These are the options we can mutate now. To not create too drastic of changes, we only mutate a residue to run similar to it according to this graph. We also ignore histidine, cysteine, proline, and glycine, as changing these will usually break a protein. From this, we optimize through a greedy algorithm where the mutation option resulting in the best improvement in CNNVS will be picked and become the reference point for the next iteration. Even if the best change in an iteration results in a decrease in performance, we will still take it. This ensures the evolution keeps progressing. Now I can just let it run for a bit. Here is our best result. It seemed to achieve significantly higher binding affinity while maintaining post-validity. Of course, our simulations are not perfect and does not guarantee the enzyme will be stronger in real life. Needless to say, experimental tests will be needed to actually put this result to production. And at the end of the semester, I presented this to the class and everyone loved it. In the future, we can adapt this project to use a better mutation and refolding system utilizing proper molecular quantum mechanics, and of course a docking tool more suited for enzymes and reactions as opposed to receptors. And as always, the code is open source on my GitHub if y'all want to play with it. Speaking of that, I got into the PhD program I wanted. It will be in full stack computational biochemistry, meaning I'll definitely get to work with the advanced algorithm and methods that expands this project. Anyways, you know how it goes. Deep fry that like button or dislike if you hate the video. Leave a comment and subscribe to support the channel. Love y'all.
Hide and seek. living the dream.